happy to introduce you Dr. Ekaterina Ivanova from Dokuchaev Soil Science Institute, and she will tell us about microbiome of the Russian soil structure, composition, and the main drivers factors. Please.
Thank you very much. So colleagues asked me to introduce the whole overview of the microbiome of Russian soils. So the main features I will introduce the following presentation. Oh my God, the same problem.
У нас по-прежнему небольшие неполадки, к сожалению.
Что случилось? It's okay? I continue? The diversity of soils in Russia is very huge, but in international projects, there were only a few samples of Russia presented by microbiological portrait, and inspired all as well by the creation of the global atlas of the dominant bacteria found in soil international. So we started our project, which is devoted to the investigation of the global biodiversity of microbiomes in Russian soils. Okay, so we formed a big collection of the samples. All the institute across Russia sent us the samples, and so the big collection was created, and we isolated the DNA from them and analyzed the microbiomes of different soil types of Russia. Their main research questions were what are the main factors shaping the composition of the soil microbiome, and how to arrange these by the influence in shaping of the microbial portrait. Does the core of microbiome of Russian soils exist? It is the conserved part of the microbiome, which is which doesn't influence by any factors, any change in land use or any soil type, and so on. And can we link the soil formation process with the specific traits of the microbial composition? For this purpose, we analyzed a huge data set of different types of Russia. There were podzolic soils, chernozems, and kastanezoms, and salt soils, solonets were also included in this data set. We saw the separation of the microbiomes of different soil types formed in different conditions of soil forming processes. So different soils were very contrast in their physicochemical composition and physicochemical properties, and it reflected in the soil microbiomes. We isolated the microbiomes of podzolic soils, and the microbiomes of chernozems, and microbiomes associated with salt soils, solonetic and kastanezoms, and the brown soils were formed another cluster. Here the representation of another factors, so we mapped it by the pH. The pH is the main determinant of the microbial structure, so we saw the clusters formed by the weak acid soils and alkalic soils. They will form separate clusters, and natural microbiomes of the natural soils formed the intermediate cluster. Also we determined some variety of plant composition So we formed the clusters of boreal soils, microbiomes, and xerophytic grasses, but maybe it's the demonstration of some other factors. So, for example, the separation of the xerophytic grasses cluster maybe is formed by the specifically climatic conditions formed in these soils, and so the separation of these microbiomes. And to more detailed, it will be better to see the influence of the plant composition on one soil type, and so we saw some separation of the microbiomes, which is specific trade patterns were determined on the different composition of phytocyanosis, so with grasses and with shrubs or with tree plants. And another representation is the shift in the structure of prokaryotic communities after artificial plantation during 50 years on the middle grass on the middle soil. It was planted large, and after it, the microbiome dramatically changed. So we saw it in diversity levels and in the composition of taxonomic composition. So we see the separate clusters of these biomes. And the planting of large are reflected in strong acidification of the profile, and we saw the more proportion of acidobacteria there, and also we saw that on the principal component analysis data, so we saw as well the separation of the large cluster and in comparison with the control soil. Sorry. And another part of our investigation was the investigation of the core microbiome. So the data set included the podzolic soils, the chernozem soils, castanezomes, and we determined that if we compare to occasional soils, so the variation is was more than if we took two random soils from one type. So they formed also the separate clusters. And only two OTUs, only two microbes were present in all of the soils investigated. They were some isolate of Solilobacteria subfamily and Bradyrhizobiotes. So these bacteria are more one is involved in the carbon cycle maybe, and another is associated with a plant community. So they were present in all the samples, but so the core is very little of Russian cells. And another way of our investigation was the metabarcoding of genetic horizon of different genetic horizons of the main types of Russian soils of the European part, and so we see the different taxonomic pattern of different soil types. So we see, and so if we see at podzolic soils and gray forest soils, alveolar soils and physomes, so we see the change in physicochemical composition and in diversity level in by the depth. And we determined that high diversity of microbes in alluvial horizons, in B horizons, due to enrichment in silt, as well as mechanical transport of cells and DNA, determines the more diversity. And more dramatic differences in the physicochemical parameters and diversity of soil podzolic soils was determined by the soil depth in comparison with the gray forest soils. And the investigation of the Chernozem soils, we see the change in prokaryotic community during transition from arable layer to the subsoil level. So as the result of plowing, so we see the dramatic lowering of the diversity. And the investigation of contrasting elements of meadow chestnut soil and solonase complex, so it demonstrates the similarity of the diversity in the upper horizons and diversity in the meadow chestnut profile soil changes with depth gradually and statistically insignificantly. And we see the change in diversity levels in solanates is characterized by the pronounced profile differentiation and sharp decrease in diversity in the solanatic, very dense and salt-rich horizon. And differences in the water-air regime of soils and soil content, and we determined meadow chestnut soil with more moist profile is characterized by a high diversity of prokaryotes, while dense solanatic horizons, slightly watered and rich with soils, unfavorable habitats for microbes. And we also determined the differences in composition, microbial composition of the different horizons of Chernozems. Here is a demonstration by two phylogenetic metrics, Unifrac and Bray-Curtis. And so we see that the formation of non-overlapping clusters by microbiomes of the A and B horizons, and the community of A upper horizons are much more distinct from each other than the communities of B horizons, alluvial horizons, mineral, which form small narrow clusters in all diagrams. And the differences in prokaryotic community structure between soil horizons were more contrasting than between land uses. So soil formation is a more pronounced factor than land use on Chernozems, for example. Another way of our investigation was to study the link between the soil evolution and the evolution of microbes, microbiomes. So is there some concomitant evolution of these types? And we investigated here the chronosequences. So we made here some change. So we used the space variants and of different ages, as the reflection of the soil evolution. And for this purpose, the soil queries, for example, non-contaminant, of course, soil queries are the good models of soil formation, and we can see and we can investigate the concomitant evolution of soil microbiome, and to see the microbial indicators of the soil formation, for example. So for this purpose, we investigated the Maluk-Sasendi query in the Leningrad region, and another Leningrad region object was Nizhny-Sibirsk Nature Reserve. And here we investigated the soil formation, the podzolic formation from the zero stage to the climax community, climax biogenosis, and climax microbial community maybe. So was the variance and the initial stage, lithostate, and to the climax soil and soils of the Nizhny National Reserve. It was the ongoing soil formation from the 70 years to the climax podzols. And we determined that the indices of the soils of Malux aquaria of the early stages of soil formation demonstrated more higher levels of diversity than the climax ones. And also in the Nizhny National Reserve, we determined the sharp decrease in the diversity of the podzolic horizon, so a result of soil formation. And here is the illustration of the divergence of the biodiversity of different points of analysis, indicating the microbiomes of different horizons. So this we can see the separation of the topsoil and the mineral part of the profile. And on the Nizhny National Reserve, the picture of the beta diversity is reflecting the soil profile of the podzol. So we see. And here is the evolution determined by the increasing of difference between the horizons during the soil formation. And the core microbiome of the Malux aquaria chronosequence. So we see that the initial stages from one stage to embryo podzol, from 0 stage to the 30 years, and the more mature soils are characterized by different, very sharp difference of microbiomes of the, for example, for dominant taxa. And also the dominant taxa demonstrate the clear separation of the top part of the profile and the mineral part of the profile in the Nizhny Reserve as well. And as we map all this beta diversity by principal coordinates analysis, so we can see that here is some gap in the stages of soil formation. So maybe we see clear separation of the first stages and other parts of the soil investigated, and maybe here is some missed part. So maybe it's the stage of 40 years. And in the all the variety of the soil formation factors, so the main factors shaping the microbial portrait, the microbial composition, are the soil moisture, organic mineral chemicals, which are referred to the soil type conditions, organic matter content, vegetation and the stage of plant endogenesis, and different seasons. So thank you for your attention.
Thank you. Now it's time for questions. Maybe 1 or 2 questions we can ask. Alexeyevich, do you have any questions?
So, could you tell, please, what is, excuse me, could you tell what is the main driving factor in soil microbiome composition? So, moisture or organic mineral, organic matter. Soil moisture is main.
On the base of our investigations and on the base of the literature data, maybe we can see we can say that it depends on the of the location of the climatic zone. Maybe
Yes.
Maybe in the north part it can be temperature, the limiting factor. And maybe
The
in the south, more south regions, it is moisture. So it depends
After
on
temperature. Moist after
Moisture is more significant maybe in the south regions. There is it's very limiting, and it's shaping the microbiome.
Well, thank you. Thank you.
Maybe someone from Zoom would like to ask a question to Ekaterina. No? So I also have one question. You presented a lot of results on soil microbiome structure and composition and so on. So we know so much on the soil microbiome. And how do you think now, where should we go now? And what scientific directions related to the soil microbiome should we consider now?
So now we should go to the functions and what should they do. So we found what are them, and now we should go what do they do, and to link the physiological patterns and maybe metagenomic patterns, so deep to the metagenomic, and to link it with the physiological traits.
Okay, yeah, that's a good answer. Thank you.
Thank you.
If anyone has no questions, thank you again, Ekaterina.
Thank you very much.